What I would like to get through today mostly will be describe a little bit what is meta science and what is the role of meta-analysis within meta science. The first task I'll then ask you to do is to come up with some questions in your own topic, what a meta science project could answer. Then going from that, I'll then go through how to do a literature search, how to find papers relevant to that question. Following that, I will then show you how to screen for papers. This process of once you do the literature search, you make it a few thousand. How do you get from that few thousand papers to the realistically tens of papers that will be relevant to your question directly? And then if we have time, I'll then go from following that literature screening to do some like data extraction and how you can actually get the data you need from each of them papers. Before I begin this properly, I'll give a preface that this really is the tip of the iceberg, and it is impossible to be really going away after a few hours seminar on how to do this and then being able to do it. Realistically, there's lots of textbooks and resources, and we can provide some of them after to really embark on this journey to really learn how to do this properly. And I think the biggest take home away message, which I can even say now, is it's really important to plan this properly at the start. Because if you do this at the start and don't plan, you may end up with too many papers to screen, too many papers that are included. And if you're in this situation, you actually won't really be able to get it finished. And I have a few anecdotal experience of people doing this, screening through tens of thousands of studies, getting a few hundred papers that are then included for their question, and then they're not never be able to actually extract the data from these hundreds of papers. So it's really important at the start to realize how much effort can I really give towards this, and having kind of the end goal in sight when you start and be able to plan it that way, because otherwise it can really run away from you. So, what can meta science actually answer, and what are different types? Simone already mentioned about systematic reviews and meta analysis. And as a framework, what these typically aim to do is look at what are the impacts of X on Y and looking at the results. The way typical questions of this could be, what are the overall effects of X and Y? You could look at how does X impact the variability of Y. You could also look at, is this impact of X on Y consistent across studies? This is typical in clinical trials in which they would show that a drug has some kind of impact in cancer. And what they'll want to know is it actually consistent across the different studies that have investigated this drug? And you can also look at what are drivers of this variation. Is it because it's in different location, using different methods, or is it using different other things? So from a systematic review or meta-analysis, there's also a systematic map or scoping review. And these typically aim to look at what people have actually done rather than what they have found. And these methods are typically used to see what are the gaps in our current understanding, what methods are typically used, what species have been investigated, and what exposures or interventions have also been used. This is typically used in medicine, but is definitely becoming more routine in ecology and evolution. And I think it's nearly went up in order of magnitude how much systematic maps have actually been done to identify gaps. Particularly, it's good if you have done a systematic map and later on you're writing for money that you've actually quantified that there is a gap here and you've got evidence to suggest that rather than saying a few people have just said that we don't do this yet. You can also do a bibliometric analysis, which actually is looking at the researchers themselves most of the time. You can look at what countries have been conducting the research, how different people interact together, what are the collaboration networks, and you can look at the main researchers in literature. We're also working on some methods right now to be able to use these results of bibliometric analysis in a statistical analysis. So we can not only show what is the impacts of a drug on an outcome, we can also show how that which research groups are driving these results and potentially identify some biases in people that they may be funded by the drug company and things like this. And this is kind of all being routine in medicine, but I think it's bleeding its way into ecology and evolution and freshwater ecology in general, of why do you actually get the outcome you're getting? Is it partially due to conflict of interest? And with bibliometrics is really the key to be able to look at that. You can also do what is called a critical appraisal, which is sometimes involved in a systematic review or systematic map, in which you'll look at the actual quality of the research within your analysis. With as with any of this kind of research, you are only as good as your included papers, and critical appraisal is the way that you're able to evaluate the quality of them papers. Unfortunately, at this time, though, to evaluate the quality of primary studies repeatedly between different users or different papers, there isn't that many tools yet. And even within peer review of experiments and stuff, everybody seems to do it differently. And there isn't that many tools yet to be able to appraise the quality of a primary study, especially in ecology, evolution, and freshwater ecology. In medicine, there's a few tools such as the risk of bias tool, and this became quite widely used to indicate the quality of a clinical trial and whether if it and what people sometimes do there if it doesn't meet a certain quality, they were actually rejected from their paper and not included, which is another criteria that people use. But I'm yet to see that in our ecology and evolution sphere. Some examples that I've asked in some of my research so far. I've looked at the impacts of pesticide exposure on fish behavior consistent across species. So, does it really matter what species is actually eliciting the behavior? The impacts are consistent or not. You can say, how do people do the measurements of behavior change the results? You can look at what pesticides have been studied and where the gaps are lying. And you could also ask, is the research in general reliable? What is the reporting quality or what is the conduct quality of the current papers? So the first task I would like people to do, which you can give 5, 10 minutes for, is to come up with some kind of meta-analysis or meta-science question for your own area. And then from this, we'll be able to push off and create some search terms and screening later on. If you cannot come up with a question, it's completely fine to use the question I've got on the screen here. But I think to get the most out of it, it would be good to come up with your own question.
There are some questions popping up in the chat.
Yeah, in regards to the first one, this is quite a good point. So I think that it is quite typical in a meta-analysis or something that you may find that the results have changed over time. And what people were finding in the 70s, 80s, and 90s is not what people are finding now. And then what you can do from this, if you can show that there is some kind of relationship with time and results, then you can start to explore why. Is it due to the different methods purely, or has there sometimes been different incentives over time? I think there's a thing, this phenomenon called time lag bias, which if you're likely to find something for the first time, you will report a higher effect than someone that will come behind you 10 years later and repeat it. And that is partly due to the incentives of being extremely novel with a big result at the start. So you'll be able to explore why that does happen. So that would work for sure. And you can also, which we can discuss a little bit later, filter the data that you're including in your meta-analysis for a specific time period if you're wanting to see what is the current research and stuff like that. There seems quite a lot of good questions which seem to be relevant. I think in general, with a systematic map, if you're saying that what is the gaps in a certain literature base, you can ask something that's a little bit broader than a meta-analysis. So I think if to put it in terms of the apple and oranges, the scoping review is to see what's in the actual basket, really. Whereas the meta-analysis, you do need to make them comparable in some way, which that can be as broad or as narrow as you feel like. It's just you need to be able to combine the results. And I've had even some cases in my own stuff, to be honest, that I've been a little too broad, and you start to see that some things are pulling in two directions, or some things are actually fogging the simple result. And that could even be done with something where I've combined two different behaviors or something like that. Sometimes they're not all singing to the same sheet, if that makes sense. Um, yeah, so the next part of that, now you've came up with your questions and you've got some idea of what you want to achieve. The first step is really thinking about how do we actually get papers on our question. So, mm, so the first step of this normally is you'll come up with what scientific literature databases are actually relevant to our question. If it's in ecology, you might not use something called PubMed, which is mainly on medical research. So what you're really identifying is where are the papers stored that relevant to my question. Typically in ecology and evolution and adjacent disciplines, people use Scopus and Web of Science. And the general consensus is that you use multiple databases, combine the results, and remove the duplicates. Because it is quite possible, which happens quite a lot, that things are on Scopus or things are on Web of Science and they're not on each other. So the overlap is not perfect, even when you use the same search terms. The only challenge with this in total is that they use slightly different languages and these things called Boolean operators. So what you'll need to do when you're doing it for the first time is looking up what terms are specific to which database and how to do this. But within the Web of Science and Scopus websites, it is relatively easy and there is quite a lot of resources on how to construct these and translate them between journals and stuff. Some of the databases I've used in the past include Scopus, which I've mentioned a few times now, Web of Science. There's then PubMed, which is mainly for medical research, but at least in what I do on the pesticides, there's quite a lot of stuff in the medical literature. These three databases typically look at already published papers. There's then also other databases to look at grey literature, such as government reports, or to look at governmental reports or thesis, which is one most widely used one's called ProQuest, which you can search for. But I'm unsure how comprehensive these actually are, but they do get hits that can be relevant, and BASE, which is run by a university in Germany. Um, when you're actually designing your search terms, I typically think in a what you call a PCOS framework or a PICO framework. And what we do here is we think of a certain population. Say in the case of my stuff, I could say the population is fish. You will then think about some exposure intervention. In the case of my stuff, this would then be the pesticides. You want to think in each paper, particularly in a meta-analysis, what is the comparator? Because what you normally do is then you contrast a control group to a treatment group. So you may have criteria which says that there must be a control and treatment. You'll think about your outcome, which in my stuff could then be behavior of some description. And then you could also filter whether or not it needs to be experiments, observational data, or otherwise. And you can also filter for time frame if you wanted to be on the most recent 5 years or do you not have any restrictions on time frame. So throughout when I will be giving a little bit more information on the search strings, I kind of structure it within this PCOST or PICO framework. And if there is no restriction on it, normally then it actually is not in the search term. So when we're thinking about these Boolean operators, I'm going to go through these are what is used in Scopus. Um, the first part will be what location are you looking for these terms in? The most commonly used will be to look within the title, abstract, and keywords. You can also look for these terms anywhere in the paper. However, when you do this, the hits can be huge because people maybe mention the word medicine or something, even though it's not in a medical paper, but it is less likely that they will say them words in the title and abstract. If you want to find a paper directly, you normally search for the DOI, in which you use the DOI term, and maybe you want to look for specific papers in a journal in which you can use ISSN and then give the respective code for that journal. So these are kind of the four main ones that I've used, but there is other ones which you can look for. Titles only, abstract only, keywords only, and such like that. In terms of relationships between the variables that you're searching, you can say something or something, in which case you would say ecology or evolution. And what this means is either word or both words have to come up in the paper or the title abstract keywords. Another term that a lot of people use is then the and term, in which you could say ecology and evolution, and which this means is both words must be present in the abstract title or keywords. And you can also filter for and not, in which the first word must be present and the second word cannot be present, in which you could say ecology and not evolution. Um, some other phrases that people use, you can put something in quotation marks, which means that exact phrase must come up in the abstract. This is quite common if people are something wanting to say anthropogenic noise, which needs to be a phrase, which may not be the same as saying anthropogenic and noise somewhere else in the abstract. Um, People can use an asterisk at the end of the word, which can mean you can have anything after that term. This is quite common when dealing with American and UK spelling at the same time, in which you could say behavior up until the the OU is in it, and you can put an asterisk so it can be either or. And a term that I use quite a lot, but I don't see people using too much, is instead of using the and term, you can use the W/N term, which means that the two words must be within so many words together, which is a little bit less broad than using the and term. It could be at the start of the abstract and at the end, where sometimes people might say, um, some word within ecology and evolution, which may change it. However, with this term, you're able to still get that without using the direct phrase. In general, you want to be able to get as many terms as you can think of and use all the synonyms within that. However, as you keep adding more and more terms, you will, in theory, get more and more hits. And this is a bit of a trade-off, is because you need to think about this is too many to screen. This word is not necessarily should not be added because it's adding too many terms. So what you really need to work out, which is quite tricky, is does adding that term make me miss? If I remove that term, do I miss relevant literature ultimately, or does it add too much noise around this? Because sometimes adding some words like frequency or adding some words like amplitude can add lots of stuff in engineering at the same time to what your actual question is. So you need to be quite careful and thoughtful when you're adding these terms. But what you can do is being quite clever. And when you add this term, you could maybe pair it in a phrase with something else. Or you can say it must be within several words of this other word that I know will not be in a different evidence base. Because otherwise, if we are just to add all the terms we can think of without thinking carefully, you may as well not do a search and just screen everything that exists in the first place. Because the search is actually to restrict what you're actually trying to sift through. And that's why when I said earlier, if you have no restrictions on one of the PICO criteria, you actually don't need to search any of it. The search is actually saying you're restricting it to these terms. What you can then do is test the sensitivity of this search, in which you could look for some papers using Google Scholar or via bibliographies of other relevant papers. And what you can do is conduct your literature search and then say and DOI and look for these papers. And typically, if you can find all 10 of these or 15 of these papers that you've thought about, you can then say that, and you can give that as a number that it was sensitive enough to detect X amount of independently found papers. And then this is a way when if you get the reviewer comment saying, how do you know your search is in any way valid? This is a way to be able to measure that beforehand. And you can say that we found 10 papers independent that we knew were relevant, independent from our search. It is important to notice the note as well. If you don't find one of them and you cannot, it may be because it's not in the database. It's not necessarily your fault, but it's just about, I think, being transparent. In the fact of saying you found 9 out of 10, you did not find this one. I think that's completely fine. It's better than not measuring it and getting the comment and not having any justification of how is your search in any way good. Um, Outside of using scientific databases, there's also a few other strategies people can use to look for papers. A way to do this is to look at the bibliographies of papers that are relevant, or look at papers that have cited relevant papers. And this is called backwards and forward citation searching. Typically, in my experience, adding this does add thousands of papers, especially if some of the big reviews, are relevant or perhaps cited hundreds of times. So I think use this quite sparingly, but I've found it quite useful to find some papers that may not be indexed properly and stuff. So the kind of way I would say this, if you're finding lots and lots of papers via this method and not too many in your literature search, it's maybe because you haven't got the right terms. And you should maybe look at these relevant papers. What terms are they saying to maybe incorporate them into your literature search? Um, yeah, and then if you do multiple database searches such as Scopus and Web of Science, you can download, which we'll be able to do later, download the hits and upload them into RDU's Rev tools, which is an easy way to deduplicate the records. And you can do this with saying they're 10% similar, or you can do this to say they must be exact title matches or whatever threshold you want. And sometimes the overlap can be very high, up to 80%, but sometimes it's a little bit lower, depending on the databases you're searching. So the next thing I would like you to do would be to try to come up with a Scopus search string for your question. I've put a template one which is quite long on the board here, which I've got some of the terms of the title abstract keywords. I've some asterisks for even after and before the words. I've got some of this within so many terms or so many words within each other. And when you're constructing the search string, one thing to note is if being careful with brackets, because sometimes one of the brackets can be off and it won't work. So it's being careful that you close your brackets and stuff like that. Yeah, Simone, you have your question.
No, I just thought it would be a good moment to interrupt. And we
Yeah.
have a question on the chat, and I also have a comment about the combination of keywords. Michelle just mentioned that what is the standard practice with regards to manually adding studies that you know would be relevant but did not come up in your search? So what would you do if you found this? You haven't found a very important paper in your search. Would you add this manually, and how will you inform people about it?
Well, if you've done your literature search, all the strings are done, and maybe 1 year passes, and now you've extracted the data and you're analyzing it or something, and you find another paper, it's up to you in terms of you can just include it and just put it in the flow chart. I have found some extra papers, but I think it's careful to note that you did the literature search in a certain day, and there will be an extra year of research than from the day you do your search realistically for the paper coming out. So it's up to you whether you're willing to haphazardly come across papers in that year and you'll include it, or you could say it's not. What I've typically done is say it's not published in the day I done the search, I won't include it unless if I come across the paper and it is before the date, I would then include it and just note that this was found external to my literature search.
For example, sometimes we have papers published in some journals that are linked to societies, and it's not available in Web of Science or Scopus. And they are very old papers, for example, very seminal papers. And Michelle, if you check the flowchart that Kyle is mentioning, the Prisma flowchart, we have ways to inform people that how many information we are getting from the seminar, the papers we retrieved with our research in Web of Science and Scopus, and from other sources. You can also inform this in the flowchart. It came from another source, or a database is from a research group that is important but wasn't published and you couldn't find in your search. So that's a way that we found to inform people where it came from and to include in our search. And
And
I don't know if
just
Kyle wants to.
yeah, and just to add to that, with if you know that there's some specific society journals that are not indexed and they're important, that's kind of you up to as the the meta scientist or the person searching to know that these relevant ones exist, and you should also search for these databases and stuff. I've mentioned Scopus and Web of Science because they're generally applicable to anybody, but there's a few, even in my stuff, pesticide specific ones, or there could be multiple language databases and stuff like that. Um, what I'll do now quickly is I can pop on to Scopus and be able to show you where to put the terms and stuff.
And while you're doing this, I think Mariana has a question as well.
Yeah, there are a few more questions in the chat. But I was thinking about this adding information that maybe it's not about of the time after you've done the search, but it might be that the keywords are not, or the terms are not catching information that is not in the abstract or in the title. But still, that paper is super important. And then you may have expert knowledge, like you as an author know that there are some papers that are relevant that are outside, or a co-author knows, or somebody you can ask, am I missing a relevant paper here? And that's a kind of snowball, right? But it's not exactly this back search or fourth wire search. But there are some questions about the use of Google Scholar as a database, for example.
Typically, you can use Google Scholar as a supplement to using other databases, but in general, every time you use Google Scholar, the hits are different. So it's not reproducible. You might search something one day and come back a week later, and it's not the same hits. Where the beauty, I suppose, of these scientific databases, it's the same every day, and you can actually say, I done this search on this day and get that exact pile of papers, which is then reproducible. So I can go through quickly here. If you do this is the search terms which are on the slide. If you go to the advanced stage, or you can use the basic if you don't want to type in title abstract keyword. And you do the literature search. And the brackets may be wrong and it'll come up something's wrong and you need to work out where the bracket is. It'll pop up all the hits. So this search string on this day, doing it today, got 1,700 hits, which is actually already 200 more than when I did the search a year and a half ago, which is quite interesting. You can filter by different subjects at the side, different article types, what languages they're in, is it open access and stuff like that. What you can also do is sort by year, what's the newspaper's relevance, based on what ones relate to your terms. And when you first conduct your literature search, a good idea is to conduct the search, look at the relevance and say the first 10 papers relevant to your question or not to make sure that you're semi-relevant. Once you think it's a good search, you can select all and then export, which you can export as a CSV file if you want to be able to open it in Excel or something. But Ryan, which we'll use a little bit later, likes these bibtex or RIS files, so I would download both the CSV because it's easy to use and be able to open, and the RIS file because it works in Ryan quite easily. Simona, you have your hand up.
Thanks. I just didn't want to interrupt you, but I know it's very dynamic. I just want to comment on Mariana comments earlier that she mentioned that maybe the problem is regarding our keywords. So when we are planning our search, it's important to read a set of papers that we know are related to our subject. For example, the very important ones are the ones that test the theory or brings a lot of results. So we read them fully and we make sure we are taking notes about the keywords they are using. And I also advise my students to read the references they cite. So we take a lot of information from these example papers, and with those we have a better idea of what kind of terms we can use to create this search string that Kyle is showing us. And I learned another strategy is that, for example, if you have a set of papers talking about the subject you want to um, carry a meta-analysis, for example, you can use an R package that can extract the most important keywords, and then based on the frequency of the words, how many times the words are used in the paper, it builds up a list with the most important terms cited within those 10 or 20 papers. And with this list, you can tickle around and play around with the keywords and synonyms to build the first version of the string, the search string. It takes a while. It's not something that we come up like easily. It takes days. You read more about and you think about the synonyms, and we test different combinations of keywords, and we play around if it's too complex or too restrict, and if it's answering the question we want to answer. And once we feel confident about this combination of words, then we go back and we start searching as Kyle is showing us. So here we are just trying to summarizing things and showing you the big picture, but it takes time. So don't be too anxious if you don't come up with the search string at once. So you need to read a little bit more, be confident about it, and use some tools to help you out with this. And another thing that came out on the messages here is that Justin mentioned if we can exclude review papers when we are conducting our literature search, and I'll let you to answer this one, Kyle.
Yeah, in general, yes, you can if you know that there's a specific type article type that can be excluded. You can exclude that directly in the search string. However, sometimes some weird stuff happens and it may be indexed as a review when it's actually not. So typically I have excluded that in the next step in the screening stage, doing it a little bit more manual. But I wouldn't, if I was reviewing a paper and someone had done that, I wouldn't have a red flag in any way to say you shouldn't have done that. But typically I haven't done that directly. But you can just say and we filter for article type, article not review, or something like that. Also to add, to what Simone said before, when as to create these strings realistically is an iterative process, and if you'd be able to even have step one of this today is pretty fantastic. But it's good to record the journey of the search string along. So you could have this is the first attempt, this is the third attempt, this is the 10th attempt. And in some of my papers, I've then actually provided this search string development in the protocol because then I can trace back if somebody has a comment in some way, why didn't you include this term? If I had thought about it along the full journey, I could say, well, it added 2,000 hits and that's just not, I can't do that. Or something like that. You can actually have evidence of what happened when that was included. Because I think regarding the search strings and peer review comments and stuff to get it published, if people do have harsh comments on it, it's very hard to fix at the very end, and that's something that's quite tricky. So it's all about being, have you thought about everything? How do we include, like, show evidence you've included it and why you excluded it and stuff like that. So, for those of you that have been able to conduct the search in Scopus, once you've done it, if you hit search and if you select all, then you can export the hits as a RIS file, which I'll then use in the next section. If you have not been able to do this in the email you received previously, there should be a folder called search. And if you open that, and there should be then the hits from my literature search are already provided there, which you can download and we'll be able to use a little bit later in Ryan. Stop sharing. And then I'll share the slides. Can everyone see my screen okay? Cool. So once you've done your literature search, you've now got a pile of papers which your potentially relevant studies are within. What you then need to do is to be able to find the actually relevant studies in that pile. Inevitably, when we do searches, every paper that we are in our literature search is not relevant. And this is mostly because when you're doing searches, the terms you use can have multiple meanings at the same time. And then you'll get studies that are not relevant. If you were to do your search too narrow, you will have less studies to screen. However, the chances of missing studies that are potentially relevant become quite high. So it becomes very challenging to get a very sensitive search that does not get that much great number of hits. So you end up with roughly a 5% success rate of what is actually in your literature search and what you include. But sometimes it could be anywhere from 1 to 10, really, but 5 is normally roughly where I try to make it. So from the literature, after the literature search, then the literature screening, it's quite similar to the search when I'm conducting or thinking about how do I actually screen for relevant studies. I still follow the PICO framework that I would have made for my search. So the screening criteria that you normally think about is it the correct population? Is it the correct exposure? Is it the correct compare and contrast or comparator outcome and so on? When conducting the literature screening, we normally do this in two different steps. We normally look at the abstracts first, in which we see can we tell by the abstract whether or not each of our criteria are met. And the ones that do meet our criteria or could potentially meet our criteria, we then will look at the full paper and screen the full text to see if it's relevant. We can normally tell quite often by the title straight away that this paper isn't going to be relevant. And it is okay to basically read the title and say that this paper isn't included because we're looking at only fish and in the title it says it's on reptiles or something. You can normally tell that straight away. So the papers that are included in the from abstract screening are then we then look at the full paper. There's quite a lot of debate around this, even in my own lab, is whether or not the screening should be done in duplicate by two people at the same time. The benefit of this is if somebody makes a mistake, it is quite possibly going to be caught by the other person. And when you do this, then you'll have the two people, and if there's a conflict between them, they then can discuss and resolve it. However, to actually do everything in duplicate is very resource intensive. You need multiple people, and you need people that are willing to give quite a lot of time to this screening. Typically, what I've done when I've done this, I've maybe done 80 or 90% of the screening myself. And then each of the authors that I work with do 10 or 20% each to make it in duplicate. But there is a lot of debate whether or not this process is worth it. In medicine, this is kind of standard, and they'll ask if you have why you haven't done it more and more. But in ecology and evolution, some people do it, some people don't, and there doesn't seem to be too much consensus on exactly whether or not it should be done. And I think there is scope for a paper to see what is the actual accuracy rate of double screening versus single screening and so on. Before you actually start to do the screening properly, even from some of the searches and the pilot searches, it's important to test out your screening criteria because you might actually be being too lenient on abstract, which means you have too many full text to screen, or you could maybe be too strict in which you're actually excluding papers at abstract that could potentially be relevant at full text. So you kind of need to go through this similar to the search, plan some criteria, test it out on a few papers, come back, say, I was accepting too many papers here, I'm going to have too many, and balance this. But then where it becomes a little bit complicated, what you balance it, the screening stage needs to still work with the searches. So you kind of build up this multi-layered picture of I search for these terms, which then relates to my screening. However, maybe my question's too narrow or my question's too broad, and I'm going to end up getting too many papers included. So you kind of do this in balance, which is it's quite a tricky process because if you know that you're doing this as a PhD chapter, having something like 100 included papers is a huge effort, and you need to know that before you undertake it. Because the worst thing to do is to be halfway through the extraction, or even you've just completed screening, and now you realize there's 100 papers, and you've 3 months to finish this, and that can't really be the case. So it is quite, it's almost the most important thing is to plan how much am I screening? What is the hit rate? How many hits did I get from the search? And be in this kind of balance picture. So what I'll do, the software I normally use to conduct the screening is Ryan, which allows us to quite easily upload our hits from Scopus, and it allows us to have multiple people screening on the same profile, and it's pretty handy. So what I'll do, I'll go through Ryan and I'll be able to show you some tips and tools to be able to use it. And then from that, I would like you to come up with some preliminary screening criteria, which you can test out on Ryan for a bit, and then we'll see how we get on from there. So if you've been able to log into Ryan before, if not, it's okay. I can just take you through how to what it looks like now anyway. So you log into Ryan up here with your email address and stuff. You can then create a new review in which you can add any title for this review. You could just say pilot one. You can tell people what kind of review it is in here, which will determine some of the defaults Ryan set, but I normally just put not applicable and do it all myself. You can then put some of the domain with it, and again, this is for some Ryan defaults, but if you put not applicable, it'll be a blank slate, and then you can add some notes for Ryan. I'll just say past. Okay. Not applicable. Not applicable. And create new. Once you then hit create, if this loads, hopefully. Can you see my screen again? Is this still working? Good. So basically, if you hit create, it then lets you upload the articles. I'll try to do this again, or you can hit skip and I can do it from in here. So once you have hit create and you hit create, you can hit skip for the upload at that stage, and you can then allow you to invite people and you can skip. But once you have done that, it'll open a profile that looks like this, in which case in the overview section, which is the primary bit, you can add the references from device. And this is where you can upload the hits that you've exported from Scopus. In the folder that I've used, it would be, if you use this RIS file or Research Information Systems file, you can upload this to Ryan. If I do that here, it should then upload it. It'll tell me there's duplicates because I've already uploaded it, but that's fine. Yeah, and once it's finished, it'll then all my files should then be uploaded to Ryan, in which I've got it duplicated because I did twice. From that, then you can go into the review data, which will show you all the papers that you've uploaded. It, as shown here, it says the two different things I had uploaded previously. In your case, it will be one, or if you've done multiple literature databases, this could be the Scopus term, the Web of Science. This will have each of the references for all of the papers that you have downloaded from Scopus. If you go to the screening part, this is the main part you'll be on, which you can see here, it'll give the title of the paper. It'll then have the abstract of the paper, which then you can read to say if it's relevant or not. It'll give a link to where the paper is, what file when you uploaded it is it from, and so on. What you can then do from here on the right hand side is say some good terms to highlight green, which are relevant. So say in my case, you could do fish and pesticides, and these have then came up. And what I can do is then filter this data set for all the abstracts. Let's say fish or say pesticide. And we had a point earlier about filtering for reviews. So typically I screen them out and I'll put an exclude term here saying review, in which I can filter this database for all the times review. And I would then screen these manually. Otherwise, you can filter by type of publication, but I know a lot of the times with the meta-analysis and stuff, it's not actually in the journal as an article type review. It's normally as a research paper, and that differs from journal to journal. So what you can do is then add some good terms of things that you like for your question and things you don't want. If I stop filtering the review, and if I go a good one, say fish, when people mention fish, it'll then come up in green within the text. Here's one that has said that this vitamin B12 interacts with some pesticide on zebrafish. So there's three things that can be potentially relevant to my question here. If I think that this is relevant or I should check it at the later stage, you can say include and it'll come on to the next one. If you're not too sure, you can say maybe and so on, where you can say exclude if you don't think it's relevant. What you can also do here is say the reason for exclusion. You could just say that it's the wrong population according to my PICO framework and apply that, and that will then tag that with that wrong population. Simona, you put your hand up.
Just one thing I would like to share that in the past we used to use Excel files and screening everything in Excel. It's horrible, people. I can tell that using this one is much better. And we have a free version. I think we can have a 3 meta-analysis at once, isn't it, Kyle? So you can work in 3 meta-analysis at once without having to pay the subscription. And another thing that I think I missed, Kyle, is because you already start screening your papers, but before we have to remove all the duplicates. The same paper that came from Scopus, for example, and Web of Science. Some people do it beforehand in R or in Excel, if they manage to use Excel. And then later, after removing all those duplicates, then they come to Rayan and start screening. But
yeah,
Kyle, I want
should
to
add.
hear from you.
Yeah. So if you want to, I can send around the R code in a further email about how to deduplicate exact matches. But sometimes when you do it exact, some things might have uppercase and lowercase, and R will not detect that. So what you can do here, but this will take 5 or 6 minutes, so I won't hit the button. But in Ryan itself, there's a feature to detect duplicates. So once you've uploaded them, you can click detect duplicates and you can put in it'll highlight what overlaps between two articles and what differs. But it takes a few minutes to do and it'll load. So this is a kind of secondary way as well to detect the duplicates between databases. So what I normally do is deduplicate exact matches in R, upload it to here, then run Ryan's deduplicate, and then from that, then start screening. And another thing I'll add here, while I'm on this page, you can invite members. So you can add the email of the people you want to be on board. You can say what you want them to do. Basically, reviewer means they can screen, and viewer means they can't do anything besides see it. For some reason, they've now gated where you need to pay for it to do a collaborator where they can actually upload and delete files, but normally only one person needs to coordinate that. What you can also do, which you can do inside Ryan now or in R, you can basically then tag who you want. I think because I'm the only person in this, it won't be there. But you can add sections for what papers you want which people to extract. And you can also filter this, which is quite good for how many people have screened how many papers, and you can stay on top of each of the numbers. And in the overview bit, it will also tell you how much screening's been done. So basically, out of the 3,000 papers here, we've only screened 3 papers, and it says how long I have had it open for and been able to do that for. Yeah, Simone.
Sorry, let me know if it's too much. I'm
That's good.
just
That's
trying
good.
to interact with you. And another thing that we usually have is doubts. So many of the students, they have many doubts. Oh, is it valid or not? Shall I include or exclude? So at this stage, it's important for you to separate these papers you have doubts. And later, if you don't have someone else helping you with the meta-analysis, talk to your advisor or to a colleague that is working in a similar area. Show them your inclusion and exclusion criteria, and then think about it together in order to decide if it's going to be included or excluded. And don't do it like exclude before talking to other people or thinking more about it, because otherwise you're going to have to like redo things and it's too much work to come back and do it again. So separate them if you have doubts, and then you decide later and keep moving forward. And if necessary, if at this point you find out that you missed something very important in your inclusion criteria, you should rethink it. And if it's in the beginning, you still have time to fix it. Now it's the time to fix. Don't move forward without making sure your criteria are working fine. Otherwise, you're gonna finish your screening with many doubts, and then you're not going to be confident to summarize the results and compare the results. Isn't it, Kyle? And tell us,
Yeah, no,
how does it feel
yeah,
for
I
you as
think
well?
yeah, I think it's quite important. To go back to the point of planning, being able, if you pilot quite a lot before you start, you'll have seen half of these problems before you've really committed to it. And I think it's really important to pilot, see the issue, solve it, and do this process a couple of times rather than getting halfway through this pile and then realizing there's a few issues. Then the next step would then be to try to come up with some screening criteria. What populations would, from your relevant question before, what are the intervention or exposure or the question? And then if you could upload either my file or the file that you've downloaded from Scopus, upload that into Ryan and have a bit of a play around with it. And I can maybe give, I'm just aware of the time, so 5-10 minutes to see if you can get that going. And then I'll have some just closing slides and we can have some questions after.
And I would like to ask you, if possible, if you could show us an example of your PICO or PICOS for one of your studies, and how did you manage to find the right combination of keywords?
It's a good point. I can see if I can share. So in general, I would present my Pico stuff either in a table that looks like this, which would be a screening flowchart, in which case I can see if I can get an actual one. Yeah, so normally I would put a Pico table or the Pico with the Pico flowchart in supplementary material of the paper. So the supplements end up being quite long, so I can show one. This is from an older paper I done a couple of years ago. Share. So what you can see in the left-hand side here would be the flow chart, which I followed to do the abstract screening. So I basically said here, it must be an experiment. It must be on an adult life stage. It must be pesticide and it must be behavior. And I followed that um, order of rejection. So at the full text stage, I didn't say this, we normally record what the reason for exclusion at full text and give that in the paper. At abstract level, there's too many, and we don't normally do that, but you can do that if you want. So this way, you would have the exact reason it was excluded. I excluded, for instance, a paper at step 3. I can say that's the pesticide, and that's quite clear. I also create the similar thing for full text, which largely mirrors the same thing. Just you don't. It can't be maybe. It must be correct. And sometimes you might add some layers that you can't tell at the abstract, but you can tell in the full text. Sometimes that could be the life stage that people don't mention that, or if you're doing some, it must be at a certain temperature or something like that. That mightn't be in the abstract. And then I also have what you call a pico table, which looks like this roughly, which I basically just describe in detail the relevant population exposure and stuff for this study. And then as you can see here in the next bit, I then provide all the articles I excluded at full text with the reason and the DOI for that paper to say this is the paper and why I excluded it.
While people are trying, I have another question for you. Why, even after having a very good combination of keywords based on the PICO and the PICOST, we still have lots of nonsense papers coming out of our search. And why is this always happen? And how can we prevent of this to happen?
It is quite tricky. It's mostly because words can mean multiple things at the same time. And it's actually incredibly surprising what's picked up when it shouldn't be. So I think in general, what I've kind of learned over time, just if you can get it in that kind of 5% acceptance rate, that's actually very good. And if you're having much higher than that, you're probably not getting enough hits and you're missing a lot. So it is tricky to be able to even get your head around this that only 1 in 10 of the papers I'm putting in are actually relevant. But that's quite typical because I think you need to still search quite broad to be able to get all the potential relevant papers. And then within that, you can, well, if you search exactly for what you're looking for and if you had 100 hits and you had a 95 included papers, I would argue that you've definitely missed. There's very unlikely that you've somehow got that 95 papers.
But for example, sometimes when we are searching at the Web of Science or Scopus, and we want to find things related to environmental sciences, and then suddenly we have something that belongs to like material engineering or something like that. And my students they were wondering, why do we see this kind of things? And I told them that sometimes the paper has the combination of the words, or they use a study case or something like that, and they use those words that are common for the environmental science, but the paper itself doesn't mention anything or doesn't bring results that are important for us in environmental sciences. But when the search string are used in Web of Science or Scopus, they are comparing word similarity, but not the context. So that's why we need to screen the papers and we need to read them, because they may have the words we asked in our initial search, but they are not talking about the subject we want to include in our search. So that's why we humans are still important, because we can differentiate between what is a similarity between words and the context that we need to use.
I think I was asked to share my screen again. Is that what Mariana said?
Yeah, that's a question to share. I love that phrase. We humans are important.
Yeah. So, if you've been able to then upload your papers, I don't really have a specific end goal with Ryan, but if you have what you would like to include in your question, or even any question for that, my question would be just to have a play around. You can have a look at, these are all the articles, the ones that I've had a decision for, and the ones that I haven't, and just be able to add a few include words, a few exclude words, and play around with the buttons and stuff. And then what I'll show when I'm here, once you've done the screening, you can hit these the three buttons here, and you're actually able to export this file, which then you can filter for the included files. So if you hit this is all undecided, but I can go here and say I want only the included files, which will be one. I can then go over here, export filtered, and this will be that one paper, and I can get the decisions, and which I can easily export that, which then can go on to the full text stage, or export that because that would then be the final length list of papers that I'll then be extracting data from. Yeah, so what I asked to do, if you can just kind of come up with the screening framework, in which case think what's your relevant population and what's your relevant exposure, and so on. Yeah, because I kind of always thought about these things in this kind of flow chart, which is kind of a binary yes, no decision. And if it gets to the end, then it goes on to the next stage. Because this is, I think, it's the easiest way to record why you actually excluded it. Because in theory, you could have someone comment, why is my paper not in your meta-analysis when I think it's relevant? If you do this, I can actually have a reason for that. And maybe I made an error, maybe I didn't. I can have the reason that maybe the study design didn't fit it because they didn't have a clear control treatment, or the control and treatment with the same individuals, and I didn't want that, or something like that. Cool.
And Kyle, it's important to highlight that we can only do this step if we are very confident we have a good question or we have a well-designed question to be answered. I'm just gonna give you an example. One of the students, they wanted to understand the carbon budget in aquatic reservoirs, and they wanted to know how the carbon budget would vary depending on the vegetation around the reservoir. But then it was very complex and it was a very big question. And then they just found out it was very big and complex when they start to include or exclude the papers, because they then came to the idea that, okay, we can't compare for all the forests. We are only comparing the ones that there's riparian forests around the reservoir. We can't compare carbon budget in the water and in the sediments. We need to think about it beforehand. So it takes time if you think about your question a lot and discuss it with your partners and colleagues and your advisor. So then it's gonna save you time and makes you more confident when deciding if a paper will be included or excluded. And that PICO strategy is very important because are you going to talk, for example, in this situation about all the reservoirs in the world or just the reservoirs in South America? What is considered not vegetated or without forest? Is it a bare soil? Is it with grass? And then you come across this kind of questions that you don't need to solve them afterwards if you have them well discussed beforehand. You have a very delimited and sampling universe. And what is your population? All the reservoirs in South America, they need to be compared to a reference area. So treatment and control, or treatment and reference. Or no, I just want to synthesize the effect without comparing it to a control or a reference area. And what I'm going to extract, is it the carbon rate or the carbon content in the water or in the sediment? So it takes time for you to develop the strategy and the combination of the
This is a
words.
process of knowing what's your being very specific, what is your question, and how many papers are you actually willing to get at the end goal, because you can narrow this at the start if you just were able to foresee it. And I think that's quite tricky. So I'll present my last couple of slides here, and then if there's any burning questions, I think that's that. Let's see. Cool. So I also planned some things which we don't need to do now, but if you were able to find any relevant papers, and there's some resources also in the other folder, which you can also extract some of these relevant data for your question. This would be such things as trying to find species in that paper. It can be what is the duration of exposure, it can be the study design setup and this kind of thing. So there's some in the folder of extraction. I've got a table there which I've extracted from a paper before. So what you can do, if you can find a couple of the papers um, in Ryan that you think are relevant, and you can also then play around with after um, about how to extract the data. So just to finish up kind of on everything, I have presented this in a way that makes it look quite, you do the searching, or you get a question, you search for it, you screen for it, then you extract for it, and it looks like a process that is quite linear and one after the other. But in reality, it's nothing like that. And in reality, you will have designed your question, you'll have then searched, then you'll have a problem with the search. You need to go back to the question and change it, which then changes the search. And you layer this up, then eventually you do the screening and you conduct the screening in some way, and then something will change there, which means you change your search, which means you change your question. And I kind of do this in what I would call a pilot phase, in which you've got these cogs and you're constantly changing them to balance them to get something that you can actually work towards. And then once you've completed that pilot stage, I would then advocate for pre-registration, in which you can put your plan on OSF or the collaboration of environmental evidence or something, in which you can, here is my plan, what I plan to do, here's evidence that it works based on my pilots. And then once you submit your protocol, then you can basically say, right, now I'm going to do it all for real because I've done it all in little clusters of 100 papers or something like that for the screening, or I've extracted 5 or 10 papers and I know that this works. Because I think that's the only way to develop. How do you know what to extract from papers if you haven't tested it and you don't know what's in the papers in the first place? You can only do that by reading and practicing. And the same with the screening. How do you know how many papers that are relevant when you're actually screening if you haven't went and done it for a few hundred papers? And the same with the searches. It's do the search, see how many you get, and think there's too many, potentially too little. Need to reevaluate. But with that, in turn, when you reevaluate, you're changing your question, you're changing the screening, and you're changing the search at the same time. So it's all about this balance between the three things and the extraction too. Because if you know that you have hundreds of papers, you might need to extract less from each paper, which means less insights. So kind of just to go back what I said at the start, in conclusion, is this can get really big really quickly if you don't plan properly at the start. And the only way to be able to plan properly, I think, is by piloting, which essentially what we've went through today is a short first go at a pilot of, I've got a question. I tried to search for that question. I had some idea of what I wanted to include and exclude and tried it out. And then if you go on to do it, you could pilot some extraction of what you want from them papers. But once you've done this step, really you need to go back to square one and do it again. And this process can go on for a few weeks, really, until it's in a place where you think it's good to go. And I know I can answer the question, because the last thing you want is you've planned to extract all this data from 100 papers and you realize I don't have enough time or resource to actually do that. And then you end up doing something that isn't systematic or doesn't look that good, where you say, I'm just going to half the data set or something, where you could have really planned for that and wrote that was going to be the case at the very start if you planned it. And with that, thank you. And if there's any more questions.
I think it's worth, I just would like to ask Kyle to tell people why we didn't go to the statistics. We stopped at this point.
I suppose the reason I didn't go into the statistics of it, I think to go from step zero to that step at the very end would have been even more overwhelming. And I think a lot of this kind of stuff needs to be done in person, and to really understand the statistics is a textbook job. And you can go through how to run the model and stuff, but I think it's actually more important to understand how to construct that model and how to get the information that goes into it in that way. Because I think some of the coding in general is a little bit plug and play. If you know if you know your question, the packages are well enough, like metaphor, are well enough written to run the model. And there's resources to do that a little bit more. And I thought it was good to do this part.
There's a question from Maria. I think Maria, you can talk.
Hi, can you hear me?
Yes.
Or do you hear me double? Or it's just me? Oh, you hear me well. Okay. Thank you very much for the talk. I just wanted to ask how many papers normally you can find in the first search, because I can see that you find like more than 1,000 articles, but actually I don't know if it's normal to find, for example, around 300 or 250, or this is something that it looks like something that you have done not properly or wrong.
No, that's a good question. Typically with 250, 300, you can get that for two or three reasons. Sometimes, especially with your very first search, you mightn't have all the synonyms. So, for instance, if I just search fish and pesticide and behavior as three terms, you probably would get 300 or 400 hits most. It's only when you layer up the different types of fish and you layer up the different pesticides and behaviors, you'll get something into like 1,700. But to have, or alternatively, the question could be that narrow and you could only have 300 or so. But if you're only aiming to get 15, 20 papers included, having 300 could be possible. There's nothing to say it shouldn't be. But sometimes I would then think about, is there more synonyms you can add? And if you've 300, you can afford to be a little bit more generous and use quite broad terms and then do a little bit more screening. Whereas if you're dealing with very high numbers, you can't afford for broad terms, if that makes sense.
Yeah, thank you.
Thank you.
There are more questions. There's Justine saying that they have used Ryan before, and they wonder if they have used other AI data builders such as Research Rabbit or any other software when assessing literature.
I've used one called Covidence before for screening, but it's paid and it depends what institution you're using. I did prefer Ryan, but although Ryan seems to be going away where they're asking you to pay for more and more, which is a bit unfortunate. And then for some AI stuff, I've used Elicit AI, which you can ask it to AI screen for papers and stuff. And it's pretty good and accurate, to be honest, but I wouldn't feel confident in doing that as your first protocol without double checking it. Perhaps it's a very useful thing. If you know that you're on your own, use screen and it double checks you, it will definitely find some potential mistakes and stuff and it would be useful, but I wouldn't do it at least at this time and moment without checking it. And at least the consensus is if I was to peer review a paper and somebody had used an AI approach without double checking it, I would need to be like, how did you validate this and know that it's truly accurate and stuff? But it will get there quite quickly, I think, and it'll soon be consensus that you can do it and not even look at it quite soon.
There's another question saying, is it possible to design and draw the prisma using Ryan?
Yes, so Ryan, you can enter the Pico table, and I think it can now also do the Prisma flowchart. Typically with a Prisma flowchart, I've just downloaded it from Prisma's website or their paper and filled it in. Or someone mentioned Neil Hadway has a Shiny app which can populate it. But depending how artistic you want to be with the with the flowchart, if you want the very standard one that looks like that, you can use directly from the Prisma website, or Ryan can do that. Or if you want to make your own to fit the theme of your paper and stuff like that, then you can just make your own in PowerPoint or something like that.
Great. There are more questions by Maria. Can you create plots with Ryan about the number of papers by year, for example?
So I think with the paid version, they do let you do summary plots and stuff. But typically I haven't used it for that. What you can do quite easily, if you can download the CSV from Ryan, and you can do it in R quite easily. Is typically how I would do it. I'll have nearly a plot in every meta-analysis with how many papers per year I normally do.
That's an important question. Of access to Scopus is difficult, of course, in many places. And do you think Dimensions.ai is a good alternative? Or
Yeah,
any other
Dimensions can be quite good, and there's also OpenAlex can be used. I can put OpenAlex. But the thing with Scopus and Web of Science, and the reason you need to pay for them, you get access to the paper once you search it. Whereas a lot of the, say, using Google Scholar, sometimes OpenAlex is open, but using Dimensions, I don't know if you actually get access to the papers every time. It'll tell you the paper exists, but they actually be able to open it and stuff is in still like university access in some way. So I think this is like a bigger issue in science in general, I suppose. So it's even this, which is considered relatively, you can enter the meta science world resource limited, but you still need access to papers and stuff, which is then provided by institutions a lot of the time and still requires money, which is then tricky. And I think the push the thing should be open access, which you have access to and stuff, is definitely valuable and should be portrayed or pushed. But it is tricky with these databases and stuff that they get them, and institutions are the only thing that can really pay for that.
So we truly hope this has been helpful and inspiring for research. Of course, there's much more to go and practice and learn while doing. But I think this was a fantastic opportunity to have at least the initial steps and guidelines and tips on how to then later do it ourselves. So I would like to thank again Simone and Kyle for your time, your expertise, and your good vibes and share all your experience and the learnings you have done through painful process, but you summarize and give us so many tips. So thank you again so much for this, and thank you everyone for participating, for finding the time at this time of the year and across all these time zones that we covered during these webinars. So it's very late for many of you and lunchtime for others and dinner time, and not I don't think anyone's breakfast. That's already past, but could be soon for some. So thank you all, and I take the opportunity to wish everyone a great end of the year and a better start of the next. Let's be a year of peace and inspiration.